Thank you, Dr. Cosgrove. A fantastic session to start the event today. I'm pleased to welcome our next guest today, Dr. Tianfu Zhang. Dr. Zhang graduated from Nanjing University with his PhD in biology. After working for over 10 years in CGT-related research production and quality management, he has an extensive background in a wide variety of cell therapies. Dr. Zhang is currently responsible for the R&D of CGT reagents for cell therapy manufacturing at Acro Biosystems, focusing on process and quality control challenges. Today, Dr. Zhang will be answering questions about the importance of simplifying clinical manufacturing for cell therapies and how to bridge the gap between academia and industry. So to start with, Dr. Zhang, please tell us about yourself and your experience when it comes to cell therapy and its manufacture.
Okay. Hi, everyone. I'm Mike Zhang from Therapies Systems. I have worked in the cell and gene therapy industry for more than 10 years as the head of research and development, as well as manufacturing in various biotech companies. As such, I have an intensive amount of experience in manufacturing cell therapies, including CAR-T, CAR-NK, TCR-T, and other iPSC-related therapies.
That's fantastic. Thank you so much, Dr. Zhang. Now, moving on to the specific questions today. Cell therapy is obviously one of the most promising therapies in the 21st century. But what makes it so promising?
Uh, the news around cell therapies is largely driven by the combination of specificity, versatility, and minimization of side effects against more resilient cancers such as relapse and refractory B-cell acute lymphoblastic leukemia. Cell therapies have reported over 80% complete response rate. Similarly, cell therapies also give hope to people who suffer from unique, highly individualized diseases caused by spontaneous mutations. For example, TCR-T therapies are designed to treat hard-to-drug targets such as KRAS G12V and G12D, alongside other intracellular tumor-specific markers, all of which are hard to target with conventional chemotherapies. On the other hand, iPSC-derived therapies shed new light on curing common genetic disorders, such as type 1 diabetes. Replacement of damaged or ineffective cells by stem cells has been a major part of research, highlighting a potential therapeutic avenue for not only diabetes, but also neurological diseases such as Alzheimer's disease and Parkinson's disease. Finally, tumor-infiltrating lymphocytes developed from individual patient tumors minimize severe side effects while showing an increasing in progression-free survival in comparison to traditional antibody therapies. As such, the current state of cell therapy is extremely diverse and has branched into various therapeutic avenues with immense potential.
That's wonderful. Thank you so much, Dr. Zhang. Now, moving on, with all the highlighted advancements in cell therapy, what do you see as different between the cell therapy research presented in academic journals versus what we see clinically?
There are significantly different factors between what is present in cell therapies developed academically versus the ones that have been commercialized. A significant factor is the need to consider regulations and guidance from regulatory bodies in clinical manufacturing to guarantee safety and efficacy. For example, an easier way to stimulate and expand T cells with a high efficacy is using a PHA and CoA. However, in clinical manufacturing, these stimulators cannot be used as they are both known inflammatory agents. As an alternative, CD3 and CD28 antibodies and magnetic beads are more commonly used, which can be easily removed. Not to mention, GMP-grade reagents are needed within a manufacturing SOP in the clinical trials. This only presents a snapshot of the differences between academia and industry. As such, a significant effort in establishing a manufacturing workflow with compliant materials can be extremely challenging, which leads to the extensive gap between academic breakthroughs and the presence in new approved cell therapies.
Fantastic. Now, for those of us who aren't as familiar with the production side, could you please tell us more about the clinical manufacturing workflow?
The clinical manufacturing workflow is vastly different from processes in the laboratory. Using the manufacturing process of CAR T cells as an example, the process can be divided into two parts: generating a clinical grade lentivirus for gene editing and expanding clinical grade CAR T cells. This can be further subdivided various for manufacturing clinical grade lentiviruses. We need to construct a bacterial bank for plasmid generation along with a high 293 cell bank. When we construct the virus, transfection of the plasmids into high 293 cells can be done, which is further purified. Moving into the next process, the purified lentivirus can be used to infect PBMCs or T cells isolated from a patient at a proper lentivirus to cell ratio. Finally, cell activation, expansion, formulation, filling, and quality control can all be done before obtaining the final product of CAR T cells. Again, I'd like to emphasize that each of these steps requires both quality control and sterility testing to ensure that there are no unwanted components in the final product.
That's excellent. Thank you, Dr. Zhang. Now, cell therapy manufacturing is known to be particularly challenging, but what makes cell therapy manufacturing so complex currently?
As you can tell from the CAR T manufacturing workflow, the entire process is vastly different from developing other more conventional treatments, such as pharmaceuticals or even monoclonal antibodies, with viruses and immune cells being the core components of cell therapies. Handling live reagents and a living final product is extremely challenging. Since cell therapies involve live cells along with viruses for the gene editing and modification, along with a long list of various protein-based reagents, the entire process and ingredients need to be screened and evaluated to ensure safety along with functionality in vivo. This puts more strains throughout the manufacturing process and increases its complexity several-fold.
Excellent. Now, we have time for just a few more questions for this interview today. Preparation for clinical manufacturing is critical, especially in the fast-moving field of cell therapy. But why is it so important to be prepared for clinical manufacturing?
As I previously said, the clinical manufacturing of cell therapy is different from academic research. It is much more complex. You must manufacture therapies according to the regulations and guidance. To ensure lot-to-lot consistency, you should develop the process carefully and do your best to avoid and narrow down process deviations. The materials used in the process will affect the quality of the final product very much. You shouldn't use GMP grade and higher level materials to develop the process of clinical manufacturing to guarantee quality, safety, and efficacy. Deviations from any established protocols or incorporations of unvaluated reagents can pose significant issues and cause an entire clinical trial to be redone. As such, our quality-based design is designed to ensure the quality, safety, and efficacy of our reagents for clinical manufacturing.
Excellent. Thank you, Dr. Zhang. Now, to finish out this interview, Acrobiosystems plays a key role in helping emerging companies and researchers bring their cell therapy research to life. But how does Acrobiosystems support up-and-coming researchers and smaller cell therapy companies looking to translate their cell therapy research?
We design our solutions for our customers that are looking to translate their cell therapy research and commercialize it. We understand that finding the right reagents can be an extremely complex matter, and that everyone's needs are diverse. No matter which stage our customers are in, whether it is in early discovery, manufacturing, pharmacology, toxicology, quality control, or even clinical stage, we strive to offer products that match their needs. For example, our pre-GMP products are designed for proof of accepted products and are derived from GMP-grade reagents. So when they reach clinical manufacturing, our GMP-grade products are ready and translatable. This solution also includes a wider array of testing kits, labeling labeled target proteins, and much more that could encompass the entire manufacturing workflow.
That's excellent. Thank you so much, Dr. Zhang, for a fascinating interview. I'm sure everyone here appreciates the insights that you have provided for us today, and we really appreciate your time. And with that, we will now be moving on to our next session of the event with John E. Millad.